Hello everyone, welcome back to the channel. Today we're going to talk about pharmacodynamics, which simply means how drugs produce their effects in the body. Most drugs work by binding to special target molecules called receptors. Receptors are usually proteins found either on the surface of cells or inside the cell. When a drug binds to its receptor, a structure called the drug-receptor complex is formed. This activates a chain of biochemical reactions inside the cell known as signal transduction, and these reactions finally produce a biological response. Not all drugs act through receptors. Some drugs work by direct chemical action. For example, antacids reduce stomach acidity by neutralizing acid rather than binding to receptors. You can think of the drug as a chemical signal and the receptor as a signal detector. When a drug binds to a receptor and activates it, we call that drug an agonist. Activation of the receptor triggers internal processes in the cell using helper proteins called effector systems and small signaling molecules known as second messengers, which help transmit and amplify the signal. Common second messengers include cyclic adenosine monophosphate, abbreviated as CAMP, inositol triphosphate, abbreviated as IP3, diacylglycerol, abbreviated as DAG, and calcium ions. These molecules spread the signal inside the cell and make the response stronger. Different cells contain different types of receptors, and each receptor responds to a specific chemical. For example, heart cells contain beta-adrenergic receptors that respond to adrenaline, and muscarinic receptors that respond to acetylcholine. The balance between these receptor systems helps regulate heart rate and strength of contraction. The size of the cellular response depends on how many drug receptor complexes are formed. This is similar to how enzymes interact with substrates. Receptors exist in two main forms, an inactive form called R and an active form called R star, written as R star. Under normal conditions, most receptors stay in the inactive state. Agonists shift receptors toward the active form, producing a response. Antagonists bind to receptors but keep them inactive, preventing the response. Partial agonists also activate receptors, but only weakly, so they produce a smaller maximum effect. The biological response depends directly on how many receptors are in the active state. Receptors involved in signal transduction are grouped into four main families. The first family is ligand-gated ion channels. When a drug binds, the channel opens or closes and allows ions to pass across the cell membrane very quickly, within milliseconds. These are important in nerve transmission and muscle contraction. For example, activation of nicotinic acetylcholine receptors allows sodium ions to enter the cell, causing excitation. Activation of GABA-A receptors allows chloride ions to enter, which calms the neuron and reduces activity. The second major family of receptors involved in signal transduction is the G-protein coupled receptors, which are membrane receptors that pass through the cell membrane 7 times. When an agonist binds to the receptor on the outside of the cell, it causes a conformational change that activates an intracellular G-protein composed of 3 subunits, alpha, beta, and gamma. In the resting state, the alpha subunit is bound to GDP and is inactive. But upon receptor activation, GDP is exchanged for GTP, which activates the alpha subunit and causes it to separate from the beta and gamma subunits. The activated alpha subunit then interacts with intracellular enzymes or ion channels, such as adenylate cyclase or phospholipase C, leading to the production of second messengers like CAM, IP3, and DAG, which amplify the signal and produce various cellular responses, including changes in metabolism, secretion, muscle contraction, and neuronal activity. There are 3 major types of G proteins. Gs increases the production of cyclic adenosine monophosphate, or CAMP. GI decree. GQ activates enzymes that produce IP3, inositol triphosphate, and DAG, diacylglycerol, which increase calcium levels inside the cell. These second messengers activate protein kinases and create amplified cellular responses that last seconds to minutes. The third major family of receptors involved in signal transduction is the enzyme-linked receptors, most commonly receptors with intrinsic tyrosine kinase activity, such as the insulin receptor. When insulin binds to the extracellular domain of the receptor, it causes 2 receptor units to come together in a process known as dimerization. This dimerization activates the intracellular tyrosine kinase domains of the receptor. The activated receptors then undergo autophosphorylation on specific tyrosine residues, creating docking sites for intracellular signaling proteins. These proteins are subsequently phosphorylated and trigger multiple signaling cascades that regulate metabolism, protein synthesis, and gene expression. Because this mechanism relies on enzyme activation and protein phosphorylation, the cellular responses develop over minutes to hours and are long-lasting. The fourth family is intracellular receptors. These receptors are located inside the cell, either in the cytoplasm or nucleus. Their drugs must be lipid-soluble so they can cross the cell membrane. Examples include steroid hormones and thyroid hormones. Once activated, these receptors act as transcription factors that change gene expression, leading to slow but long-lasting effects that develop over hours to days. Signal transduction systems have two important features: amplification and regulation. Amplification means that a small amount of drug can produce a very large response because each step activates many more molecules. This explains the concept of spare receptors, where maximum effect can be achieved even when not all receptors are occupied. Regulation occurs when receptors change their sensitivity. Continuous stimulation by agonists can cause desensitization, where receptors become less responsive, or downregulation, where the number of receptors decreases. Long-term use of antagonists can cause upregulation, where receptor numbers increase and cells become more sensitive. Now let's talk about dose-response relationships. As drug concentration increases, the effect increases until it reaches a maximum. Two important properties are measured from these curves. Potency refers to how much drug is needed to produce an effect. It is measured by the EC50, which means the concentration that produces 50% of the maximum effect. A lower EC50 means a more potent drug. Efficacy refers to the maximum effect a drug can produce, called Emax. Clinically, efficacy is more important than potency because it determines how strong a drug's effect can be. Drug receptor binding follows chemical principles called the law of mass action. A key value is Kd, the dissociation constant, which represents the drug concentration that occupies half of the receptors. A low Kd means strong binding or high affinity, while a high Kd means weak binding. As more receptors become occupied, the drug effect usually increases. Intrinsic activity describes how strongly a drug activates a receptor once it binds. Full agonists produce maximum activation. Partial agonists produce lower activation, even when all receptors are occupied. Inverse agonists reduce receptor activity below normal levels. Antagonists bind receptors but do not activate them. Antagonists can work in different ways. Competitive antagonists bind to the same receptor site as the agonist and can be overcome by increasing the agonist dose. They shift the dose-response curve to the right but do not lower the maximum effect. Irreversible antagonists permanently block receptors and reduce the maximum response regardless of agonist concentration. Allosteric antagonists bind to a different site on the receptor and prevent activation. Functional antagonists act on different receptors to produce opposite physiological effects, such as adrenaline reversing histamine-induced bronchoconstriction. Finally, there is the quantile dose-response relationship, which looks at how many people in a population respond to a drug. The ED50 is the dose that produces the desired effect in 50% of individuals. The TD50 is the dose that produces toxicity in 50% of individuals. From these values, we calculate the therapeutic index, abbreviated as TI, which equals TD50 divided by ED50. This tells us how safe a drug is. A high therapeutic index means a wide safety margin, like penicillin. A low therapeutic index means careful monitoring is needed, like with warfarin. If you want more pharmacology sessions, please like, share, and subscribe to the channel.